Hello. So, good afternoon, Philippines, and good morning from all over the world. So, before we begin, I have a few housekeeping notes. Today's session is being recorded to be available for viewing post-conference, and also we are on live stream in Facebook Live and YouTube. If you have questions for the presenter, please use the chat feature in the Zoom or can submit questions via the chat feature of the conference platform for the presenter to answer after the session. You can also use the raise hand icon during the question and answer for you to be recognized. Please put on your microphones on mute mode and your cameras off as the program is going on. Hope everybody can see and hear as well. Just send your feedbacks and we will quickly go through some points to note we are all have a seamless webinar. To start the program, I welcome you to the first seminar series organized by the Ecology Conservation Laboratory of the Department of Biological Sciences of the University of Southern Mindanao, Cabacan, Cotabato, Philippines. Thank you all for finding time visiting today's webinar. I am Rennie Jane A. Ellett from the University of Southern Mindanao, and I will be your MC or your moderator for today's presentation. I am glad to welcome Fawn, Sawadika, Dr. Tuanjit, Sritongchuay from the UFZ-Helmholtz Center for Environmental Research, Department of Computational Landscape Ecology, Leipzig, Germany. She will be presenting pollination services in Agroecosystems, case studies in Thailand. On our list, we have diverse registered participants for today, around 110, composed of both undergraduate and graduate students, researchers, and faculty. May I have the honor to acknowledge the participants as according to geographical locations and their institutions. Firstly, from the land down under, Australia, of the University of Southern Queensland. From the place of the famous Petronas Towers, Malaysia, University Malaysia, Terengganu. From the land of famous pagodas, Myanmar, of the Forest Department. And of course, the Philippine participants representing Luzon from the Cavite State University, from Visayas State University, and majority from Mindanao, mainly Ani Foundation of the Forest Honeybee of Ube, Mindanao State University, Iligan Institute of Technology, Davao Doctors College, Cotabato Foundation College of Science and Technology, Sibagat National High School of Home Industries, Senior High School, the Southern Philippines Agribusiness, Marine and Aquatic School of Technology, Northeastern Mindanao State University, Notre Dame University, University of the Philippines, Mindanao, Notre Dame of Marbel University, Mindanao State University, General Santos City, General Santos Foundation College, Incorporated, Philippine Rubber Research Institute, Department of Education, University of Southern Mindanao, of course. So quite lengthy for today for the list of participants, but we are all delighted of your presence in this webinar. Welcome. At this juncture, Angelo Agduma will give the rationale of the nnect. Sir.
Hello, good afternoon. Good day to all of you. So let me give you the rationale for this seminar series. So one of the key manifestos of the Ecology and Conservation Research Laboratory is to become a gateway for international collaboration across different networks around the globe. This is to bolster the scientific visibility and academic standing of our department and USN. To do this, one of our undertakings is to link our institution, including our students, to different experts and peers in the field of ecology and conservation by exchanging ideas and expertise. On behalf of the EcoCon lab of our department, I am pleased to inform you that we will be steering the nnect, the official name of our seminar series, organized by the EcoCon lab via Zoom and Facebook streaming. nnect will serve as a platform for firsthand or the latest information from experts in the field. This is a bi-monthly online seminar related to ecology, biodiversity, evolution, and conservation biology, delivered by our invited experts from different institutions within the country and abroad. The talks are expected to be participated by not only USM and the BioDep community, but also colleagues in the field from other institutions. We welcome you all to the nnect seminar series. Thank you.
Okay, thank you, Jilo, for enlightening us about nnect. Surely through these initiatives of the EcoCon Lab, a think tank, we can set the avenue for sharing ideas, connections, collaborations from all over the globe. Okay, are we all excited to hear the presentation of the resource speaker? Hold our excitement for a moment, for we will be knowing more about her through the introduction to be given by Chrysler Tanaldo. Sir?
Okay, so good afternoon, everyone. So I'm really happy to see a lot of people around coming from different institutions. So to introduce to you our speaker, first I would like to thank you, Fon, for accepting this invitation from our lab to share your expertise. And it's truly my pleasure to introduce you today to our very diverse audience. So Dr. Tuanjit Sritongchuay, or we call her Fon, is an expert on studying or exploring plant pollinator networks in the tropics. So Dr. Tuanjit Sritongchuay, or we call her Fon, earned her PhD at Prince of Songkhla University in Thailand, where she explored the plant pollinator networks in fruit orchards in southern Thailand. After finishing her PhD, she moved to the Chinese Academy of Sciences in southern China for postdoctoral position. So we were lab mates during my PhD and during her position and during this time. So her project during her postdoctoral work, she expanded her work on plant pollination interaction studies from China to Southeast Asia. So Fon mainly works on the area of community ecology and pollination biology of plants in the tropics. And she pioneered many important pollination studies in Southeast Asia, which become significant in our understanding of plant-animal relations in the region, which we'll share and discuss with us shortly. So Fon is currently a Humboldt Fellow and a part of the UFZ, or the Helmholtz Center for Environmental Research under the Department of Computational Ecology in Leipzig, Germany. This time, she is working on the exploration of a complex relationship of a changing landscape and pollination network in a global scale. So without further ado, let's give a virtual warm welcome to Dr. Tuanjit Sritongchuay. Fon, the floor is yours.
Okay. Thank you, Chrysler, for nice introduction, and thanks the department for having me for the seminar today. In my today presentation, I have two parts. In the first part, I will share how I developed my interest in pollination ecology study. And then in the second part, I will share some of my research. Okay, so first of all, I would like you all to close your eyes And so please close your eye for a few seconds and picture a scientist. Okay, now open your eyes, and I'm sure some of you picture a chemist or a molecular biologist who work alone in the lab. That's what most of people think about scientists. And the same with me when I was younger, like about 20 years ago, because scientists are making one of the biggest advances in our society. They're solving things like global warming, medical breakthrough. But for me back then, these things seemed to be too far and I didn't fit in. But I'm here today to share my story about how I discovered a passion for ecology. So this is me 15 years ago. I was pretty normal young girl who grew up in countryside with all of fruit trees and cows. And when I was in high school, I got good grade in mathematics and science. And my teacher said, you shouldn't become a scientist. But for back then, I had no idea what scientist was. And then I went up to Prince of Songkhla University in southern Thailand, which is the big deal in my small village. Actually, they really announced it in the loudspeaker. And when I got to university in the first year, I had no idea what to major in until the end of my first year. I went when we had to choose the project for internship. Of course, I would not choose any project working in the lab. So I chose to work with Dr. Sarah Bamrungsi, who studied pollination ecology of durian, which is favorite fruit for many people, at least for me personally. And from the project, we learned how to observe the flowers, count the number of bats visiting to durian flowers during the night, climb the tree to count the number of fruit sets. And I found it was so cool and it was so much fun. And from this project, I learned that science is skill set to solve the problem around us, and we can make significant impact by using it. So this is beginning of my interest in pollination ecology. I found pollination subject is great, and from this project, we learned that without bats, we have no durian fruits. And after that, I continue working on pollination ecology. I conducted my first project on pollination ecology during my 4th year of undergrad. I studied the pollination ecology of Oroxylum indicum, and I guess it's called Pinka pinkahan in catalog, which is common pioneer plant species in Southeast Asia. And in this study, I investigated what time the flowers open and what are the main and most effective pollinators. I found that the flower open from 7 to 8 p.m. and more than 80% of animal visitor is cave nectar bats. And the peak of visitation is from 10 to 11 p.m. I also did pollination experiment.
Iduyan na lang siya.
And found that the number of fruit sets from open pollination, which was allow the pollinator to visit freely to the flower, was not significant different from the treatment that we did pollination by hand by using pollen from different trees. While I collected data for my undergrad project, I found that these plant species have specific stigma closure characteristic. The stigma close within 3 to 5 seconds after touching. And then during my master thesis, I studied what are the causes and the consequences of the stigma closure of these plants. And then I found that the stigma closure in these plants help to prevent the stigma clogging from too many pollen grains on the stigma surface. However, for my PhD, as we know that both habitat loss and fragmentation contribute to local population extinctions, and this can also affect pollinator populations. And I thought if I study pollination ecology of one plant species, then we can save only one plant species and a few species of pollinators. So for my PhD, I expand my project to community level, which I study plant pollinator networks in mixed fruit orchards, mixed fruit orchards is very common in traditional Southeast Asian village life. Usually one garden, there are more than 20 species of plants, different layers of canopy, including rambutan, durian, mangoes, and mangosteen. Habitat loss and fragmentation cause lack of habitat connectivity. And we assume that forest remnants are the sources of pollinators for agricultural crops. The pollinators with the small home range will limit and only reach to the flowers within their home range. Whereas the plants in the area that far from the forest can be pollinated with only some pollinators that have larger home range. And from previous studies show that the species richness and abundance of pollinators of crop tend to decline with the distance to the forest increasing. For example, the fruit set of coffee in Indonesia relate to the number of bee species, and the number of bee species change negatively with the distance to the forest edge. Also recently, the pollination network have been commonly studied. Pollination network is an interaction of plants and pollinator in the whole community, and this slide shows the interaction between plant and pollinator. And from previous study, the landscape pattern can change the structure of pollination network. For example, the interaction density, which is the number of links in the network, change positively with the proportion of semi-natural habitats in surrounding area, as well as robustness, which show how strong of that robustness to disturbance. And they also found that interaction evenness, which is the homogeneity of interaction in the network, was affected by the proportion of urban area in surrounding habitats. So for this part of my PhD, I investigate how forest proximity affects morphotype composition of pollinator communities in mixed fruit orchards, and also how forest proximity influence pollination network structure, and to investigate the effects of landscape composition on pollination network structure. I collected data from mixed fruit orchards in southern Thailand. The orchards have more than 10 species of fruits with similar plant community, including durian, mangosteen, parkia, mango, and rambutan. And I chose 10 pair of orchards that near and far from the forest. The site that near to the forest is less than 1 kilometer from the forest edge, whereas the site that far from the forest is more than 10 kilometers from forest edge. And then for the landscape composition, I calculated the proportion of different land use types, including water, mangrove, lowland evergreen forest, lowland mosaic, urban and palm plantation within different radius scale ranging from 100 meter to 30 kilometer radius. And to observe pollinator visiting to herbs and shops, I made the study plots size 50 meter by 150 meter and observe animal along line transects. For tree, I observe pollinator focally from 4 directions using sweep net, binocular, and nocturnal and camera trap for nocturnal animals. And I found that the species richness and abundance of pollinators from the orchards that closer to the forest were significantly higher than the orchards that far from the forest. And this trend is similar with number of links and the connectance of pollination network. The networks are more robust when surrounded by higher proportion of lowland mosaic, which include tree gardens, plantation, and forest. This may be because both plants and insects in orchards that near to the forest are more diverse, providing higher resilience to loss of small number of pollinators. And I also found that the interaction evenness increased with the proportion of forest within 4 kilometer radius and urban area. This could be because the specialist pollinator are more vulnerable in urban and leading to predominance of generalist pollinator, which can cause high interaction evenness in the area that's surrounded by higher urban area. So to conclude, we found that the number of visitor flower interactions was higher at sites near the forest, as well as network robustness and evenness. The robustness was strongly positive, influenced by proportion of lowland mosaic within 1 kilometer radius, and the interaction evenness was positively affected by proportion of urban area and mountain mosaic within 4 kilometer radius. Okay, in addition to insects, some bat species are also important pollinators in tropical habitats. There are some economic plants like durian and parkia, whose pollination rely on both bats and insects. So next, I will present how the landscape composition affect on bat pollination networks. The changing of landscape and growing of urban area create different scenario for pollinator, including bats, and not all of floral resources for bats available year-round, so that the effect of landscape on pollination interaction might be stronger in low flowering season. So here I investigated whether the flowering season influence pollination network structure, especially specialization, and the landscape context affect bat pollination network in mixed fruit orchards. We used camera traps to observe bat visitation to the flowers. And then for the community level, we calculated a number of interactions, as well as the H2 index or network specialization index. H2 index show the degree of specialization range between 0 when there is no specialization and 1 when the network is complete specialized. Overall, we found 7 species of bats, visiting to 6 species of plants. And we found that Eonycteris pilia is the most abundant bat pollinator. And we found bat pollination network were more specialized during low flowering season. This could be because the low flowering season can cause high competition among bat species, then increase in specialization in bat pollination network. We also found that pollination network specialization decreased with the proportion of urban area within 30-kilometer radius. This could be, to conclude, the specialization in low flowering season network was higher than from peak flowering season, and increasing area of urban reduced the degree of specialization of bat pollination network. This could cause pollen limitation, leading to lower number of fruit set of our crops. Okay, so now we know that the landscape context and distance to the forest change pollinator communities in mixed fruit orchards. But it's also interesting to see how this affects the fruit production of our crop, which I will also present here. In this part of study, we chose study species for 3 species, which are rambutan, durian, and mango. We chose these 3 species because they have different major pollinators. In this part, we investigate if visitor richness and frequency of flower visitor and reproductive success change with increasing distance to the forest. Also, we want to know if the frequency of visiting bats and reproductive success of durian change as distance to the cave increases. Here I will introduce the study species, including floral biology. The first one is rambutan. The antithesis of rambutan and stigma receptive start at 7 a.m. and stigma remain receptive for 1 day. Rambutan is self-incompatible plant species, so the reproductive success of this plant species definitely depends on pollinators. And the second species is durian. The flower of durian open in late afternoon. Antithesis occur around 7:30 p.m. and the stigma receptive before antithesis. The corolla drop around 6 a.m. in the next day. Durian also show highly self-incompatible. The last species, mango. Mango flowers open during the night and early morning, and antithesis occur around 11 a.m. and stigma receptive before the flower open. Actually, cross-pollination is not necessary for mango pollination. And the study was conducted in the same 10 pairs of study size as previous part. To observe flower visiting animal, I observed focally from 4 sides of the tree using binocular sweeping nets and camera traps. Then we counted the fruit set from 3 inflorescence per tree, 10 trees per orchard, and 10 orchards per treatment within 2 weeks after last flower open. And we found that the number of fruits per inflorescence in rambutan near the forest was significantly higher than the orchards that far from the forest. There was also strong positive effect of insect abundance on the number of fruit set with as a linear increase. And the most abundant flower visitors were stingless bee, honeybee, diphtheria, and insects. Significantly more stingless bee were found visiting rambutan flowers near forest than far from forest. For durian, the number of fruit set per inflorescence of durian was not significantly affected by distance to the forest. However, the number of fruit set was negatively affected by the distance to the cave and increased with the bat visit frequency, while the bat visit frequency declined sharply with the distance to the cave. For mango, we found that no significant difference for fruit set of mango per inflorescence between sites that near and far from the forest, and none of visitor variables affected the number of fruit set per inflorescence of mango. We found that the main flower visitor of mango were flies and stingless bee and honeybees. So from here, the pollination success in rambutan show positive relationship with insects visit frequency because stingless bees made up more than 50% of visitors to rambutan. This result suggests that stingless bees are the major pollinating insects to this plant species. And the distance to the forest did not affect number of fruit set in durian because insects have less effect on pollination success of this crop. And again, the main pollinator of mango are fly, which is related to people house. So the highest visitation rates of bee near forest and decrease in species richness of insect visiting flowers far from the forest suggest that rainforest is source of rambutan pollinators, and the higher visitation rate of bats to durian flowers were near the cave emphasize that the role of cave as a sources of pollinators to surrounding durian. Okay, in addition to factor in landscape scale, the management at the farm scale is also key for crop production and incomes of the farmers. And next is our research that link the farm management and the economic value provided by pollinators. So every year in northern part of Thailand, the oversupply of longan during crop season is a nightmare for the farmer because the price of longan is in crop season is very low. So some farmers adapt to produce off-season longan fruits by using chemical spray. Then they can sell the fruit for higher price. However, this can be a trade-off when the native pollinator is not ready to work for them because in November and December, the temperature in northern part of Thailand is still low. So some farmers decide to have beehive in the longan garden to help with the longan pollination. So in this project, we investigated the pollinator communities and pollinator importance across 2 seasons, which is off season and in season, with and without beehive, especially Apis mellifera. We observed the species richness and visitation rates of pollinators from 3 different types of garden, including off-season without bee, off-season with beehive, and in-season longan garden. I counted the number of fruit sets at 2 weeks after the flower opened and the mature fruits from 2 experiment treatments in each garden, which are open and controlled treatments from 10 inflorescences per tree and 10 trees per garden. I found that the main pollinators for off-season Longan garden without beehive but were flies, whereas the most abundance of pollinators in off-season Longan with beehive were honeybee, which are Apis cerana and Apis mellifera. And the main pollinator in season Longan garden were white bee, such as stingless bee. In terms of species richness, in season Longan garden show highest species richness than off-season Longan treatments. I also collected data of pollinator importance of each pollinator species and found that stingless bee show the highest pollinator effectiveness, although they visit to the flower less frequent than honeybees. And I found that in season Longan fruits produce highest Longan fruits compared to other two treatments in both fruit set in 2 weeks and mature fruits. I also interviewed the farmer for Longan, for the Longan yield and price of Longan per kilogram and cost of Longan production. And I found that the farmer who practice in season Longan got highest yield. Compared, so, and the farmer will practice off-season Longan with beehive also got paid by beekeeper. In terms of cost, the farmer will practice off-season Longan have to pay more cost, but when we calculate the net profits, it shows that the farmer will have beehive in off-season Longan garden got highest income. So to conclude, the stingless bees are the most important pollinator of Longan, and it shows highest visitation rate in season Longan garden, leading to the highest Longan fruits. However, the farmer earn more income when practice of Longan season with beehive. Okay, in my presentation, we study the driver for pollinator population decline, which include land use change, agricultural practice, and farm practice, which have negative impact on pollinator communities. And this can link to the ecological functions such as pollen transfer, and all of this can also impact our crop yield and the income of the farmers. So this is the end of my presentation. At the end, I would like to thank the lab member from Small Mammal Research Group at Prince of Songkhla University and Landscape Ecology Group at Sritongchuay Tropical Botanical Garden, and thank all of Research Grant Agency, and finally, thank you for your attention, and I'm very welcome question and comments. Thank you very much.
Okay. Thank you very much, Fawn, again. I hope you all enjoyed the amazing presentation of Fawn. So it's indeed apparent to recognize the importance of insect and bat pollinators and its pollination networks for the survival and reproduction of several wild plants and crops, especially in a climatic changing scenario. And that's what we are experiencing now. So this moment, Fawn will answer some questions that you would like to raise to her. So may I know from the chat box if there are questions? So far there's none yet. So maybe we can wait for a while. May I ask from the Facebook Live and the YouTube participants if you have questions that you can ask from our resource speaker?
Okay, questions? Thank you, Fawn. Okay, we have a question.
Okay, so we have a question here. So from RD Alvarez, and the question goes, thank you very much for your presentation, Tuanjit or Fawn. I am curious about the impact of space to pollination. Can you provide more information about the impact of elevation to pollination? I heard a while ago about the lower and upper montane forests, but just an encompassing one. Thank you. So that would be for Fawn to answer.
Okay. So in my study, I didn't put the altitude to the analysis, but I did in terms of latitude, and I think it's similar. It's similar with elevation, because when it at the higher elevation, we might get lower species richness of pollinators, and also the impact of landscape might be higher when the in the higher elevation habitats. But from one part of my study, I found that the robustness of pollination network decreased when surrounded by high mountain forest. So this might be because in high mountain forest we have lower species richness, I guess. But I think, yeah, it's very interesting to investigate. And in Thailand, we don't have much higher, we don't have many high mountain. So maybe in your country, if you interested, I think, yeah, it is interested. It's interesting to see the effect of elevation.
So RD said, thank you very much for your answer, Ms. Fawn. So we'll wait for more others.
Hello, I'm Nozalipa here.
Hi,
Hello.
Zalipa.
Sorry. Hi, how are you doing?
Good.
Good to hear from you. Thank you for the nice presentation. I'm just wondering about the species diversity of the pollinators. I'm just curious, I mean, if we record a very low diversity of pollinators, but the abundance is high, do you think that shows very specific pollination system of the community?
Low species, but high abundance.
Yeah,
Low
of the
species of
pollinators.
pollinators,
Mhm.
but those pollinator visit to many species of plants.
No, I mean, in your presentation, you mentioned about high diversity of pollinators, but I'm
High
just,
species richness.
species richness. So, I mean, if we record a very low number of species of pollinators in a community or in any habitat, but with high abundance of, high number or individuals of the pollinators, is that shows, I mean, is that a problem with that?
It's not a problem if it's like that in nature.
Yes,
It's,
because I think it's, usually you measure the diversity of pollinator community, but it also depends on the plant species, right? Because some plants, they very specialized pollination system. Yeah.
It's
Okay.
also depend on habitats. For example, in urban, in the area that low species richness, like in monoculture crops. We might find very low species number of pollinator, but high abundance. So it's not a problem if we are sure that our method is standardized. So I think it's a pattern that is there.
So we don't, the diversity of pollinators is not the only variables that we measure, right? The importance variable in pollination study.
Yes, because we have to take some other value into account. For example, like how effective of that pollinator, like how many pollen grains that they carry per single visit. So yeah.
Okay, thank you.
Thank
for
you.
The answer. Sorry, I couldn't turn off my camera because I have a very bad internet connection here. Yeah, thank you very much.
Okay, thank you very much. So there is one question here, Juan, from Facebook Live, and this is from Sen Nuevo. She says, hi, Juan. You mentioned robustness and connectivity in pollination networks. Can you please explain it a little?
Okay. Robustness is show how strong of the network. For example, if one species is moved from the network, for example, in nature, one species might go extinct. And if that network is robust, which means that network will not collapse easily. But then in the network that is less robust, when some species, when we remove some species from the network, those network will collapse into small network. So I hope this is clear. And for connectance is show how it linked with the number of it relate to the number of links in the network. So when higher connectance, which means there are higher number of links in the network.
Thank you, Juan. So another question goes, good day, ma'am. I would like to ask whether pollinators' diversity and abundance is affected by diurnality or nocturnality.
Yes, I think because this I think most of plant species are pollinated by diurnal, like it flower open during the day. But this is kind of syndrome or specific. If it might be the flower that open during the night might be specific with the nocturnal pollinators. So I think diurnal and nocturnal pollinator is more related to the pollination syndrome. Like for example, bats. It's only pollinate the flower that open during the night. Diversity. If we have to say pollinator diversity, so I think during the night or nocturnal pollinator might have low diversity than nocturnal. Yes.
Okay, thank you. So your presentation really is exciting and interesting, Juan. So there is another question from here, from the Facebook Live from Merlane Unte. And she said, I've read that many bees are dying because of colony collapse disorder. What is the cause of this?
I think this is like a virus and bacteria, but if there is also landscape, the disturbance also another factor that can increase is the collapse might be faster when it's in the vulnerable area or higher temperature. But I think the main cause is from virus and bacteria. Yes. I think I found there is another.
Okay, there's another one from the Zoom chat. Juan, may I read it to you?
Yes.
Hello, I thank you for the informative and great presentation. May I ask if pollens attach to pollinators because of the pollen morphological structure or the pollinators? If we want to study pollen morphology, what would be the appropriate method and equipment to be used? Thank you. And that's from Andre Bon Flores.
Pollen, yes. Yeah, this is more like evolution, that the pollen morphology. Yes, the morphological structure of pollen will be benefit that they have different surfaces of pollen that might be different in terms of like different surface of pollen grain might have different what is called ability to attach at the pollinator body. So this is from evolution, because the plants want to get pollinated, so they shouldn't have like character that's how to have like pollinator to spread their pollen grain as much as it should. So yes, the morphological structure is important for the pollen deposition, and to study that I I took one course when I was undergrad, but I'm not the expert on this. So we did like pollen analysis. We work in the lab. I remember we did acetylysis and then look or measure the pollen grain under a microscope. But I don't have experience on this. Maybe I can send you a book or some paper later.
Wow, that would be great, sending them references for their questions. So that we will be happy about it, Fawn. Okay.
Mhm.
So are there some more from the audience who wants to ask? So there's some more. May I read it to you, Fawn?
Yes, please.
Still from Andre Bon Flores. He said, thank you, Dr. Sritongchuay. And his question is, are there any artificial pollination currently being done, and on what plants without animal interference?
Oh, I saw one paper that they used bee drone in the Netherlands. And I can't remember the plant species, but yeah, they start having that. Mhm. They use bee, like drone as a bee.
So really, pollination can be technologically aided by through a drone farm. Am I correct?
Yeah.
Okay. So we wish that there would be more questions, but on the chat box here in Zoom, there are none yet. Maybe we can ask again from our live participants from FB and YouTube. There are some more. Okay, so since there are no more questions to be asked from you, Juan, we thank you again for answering those questions and for the great presentation. So for the many questions that were raised to you, it really implies that your presentation was interesting. Okay, so thank you very much. It was a pleasure having with you your presentation, and we are so grateful for it. So at this point, our department chairperson, Dr. Florence Roy P. Salvania, will give his words of appreciation. Sir?
Hello, thank you, Ma'am Jane. So, in behalf of the Department of Biological Sciences of the University of Southern Mindanao, I would like to appreciate and to thank Doc Fon. Hi, Fon. For sharing with us valuable information with regards to the pollination system or networks of agroecosystem. So I guess most of our students have learned a lot from your presentation, and I myself also is very interested to pollination. Some of my students are will be working on pollination biology of some plants. So we might be asking some help from you, Fawn, if that's okay with you. Ayan. So we'd like to thank also all the participants of this webinar, and of course, the EcoCon Lab headed by Sir Chrysler and the group for facilitating the conduct of this webinar. For all the participants, thank you also for attending this webinar.
Thank you very much for having me, because I'm also excited to share my work. Yeah, and it's very honored to present to all of you today.
So thank you, Sir Florence. Thank you for the positive response from Fawn also. Now, at this juncture, we will give a token for as a gesture of our appreciation to our resource speaker, we are giving this token, the certificate of appreciation to Fawn, and may I read the citation. Republic of the Philippines, University of Southern Mindanao, Cabacan, Cotabato. Awards this certificate of recognition to Dr. Tuanjit Sritongchuay for sharing her knowledge and expertise as resource speaker during the nnect Pollination Services in Agroecosystem Case Studies in Thailand, held on March 25, 2022 via Zoom meeting at USM Cabacan, Cotabato, given this 25th day of March 2022 and signed by SUC President Francisco Gil N. Garcia, ABE, PhD. So let's give a round of applause to Fawn.
Thank you.
Thank you, Fawn. It's really
Thank you
interesting
too.
talk. It's good to hear again your talk. I think I heard your talk. I think it was last 2019, the last time I heard about your talk. So thank you so much. It's really great to hear again your work. And for the participants, if you're interested to know more about Dr. Tuanjit Sritongchuy's work, you can search her name on Google Scholar, and you can find her diverse work about
Okay, so a reminder to all participants that an evaluation link will be posted in the chat box for you to fill up and for you to be able to claim your certificates for this activity. So for our Facebook Live and YouTube participants, link will also be provided. Okay, so maybe before we end or to conclude this activity, may we request all participants, and Fawn is already on or with her camera, so that we can have a quick photo opportunity with the resource speaker. So this time we will be wearing all our sweetest smiles. So maybe Chrysler will do the signal. Okay.
Namado.
Okay. 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 Second page. Okay. Then the third page. Okay. Please turn on your camera. Okay. Okay.
Okay, so all sweet smiles are being captured. Then, okay, so it's sad to say that we are here to conclude the webinar. In behalf of the organizers, the EcoCon Lab, we thank you all for attending, especially our resource speaker, Fawn. We hope you have learned and enjoyed this presentation. Let us all keep connected with webinars such as this. So once again, this is Renny Jane ALS signing off, and we say thank you very much and see you soon in the future webinars. Thank you, Fawn. Thank you, everybody.
Bye.
Thank you, Fawn.
RD, salamat.
Welcome. Thank you, Fawn.
Okay, I will end na. And for all, ay evaluation, send mo na sa